Dear students, welcome to the final part of the video series on Chapter 5. This particular slide reminds us that linear chromosomes, those from eukaryotic organisms, have certain characteristics which they must possess in order to be viable and maintained within the nucleus of a cell. So those three elements are listed in the first column of this slide. The essential components are a centromere somewhere along the length of the chromosome. The centromere is a special protein structure to which the cytoskeleton can attach, namely the spindle fibers. The other element is at least one origin of replication. In this particular slide, we can see that there are two origins of replication on this particular chromosome. And finally, at each end of the chromosome, indicated in blue, are the telomeres, special sequences of DNA, which we'll mention in a future lecture. If a chromosome has all these three components, it is then viable and retains itself, as mentioned earlier, inside the nucleus. So during the cell cycle, the first stage is known as G1. The second stage is known as S phase, and the third stage is known as G2. During those phases, the chromosomes go through the changes indicated at the beginning of this slide. The final stage of the cell cycle is called M phase, in this case for mitosis. There the chromosomes are shrunk by being wound even tighter than before, and we have the attachment of the spindle fibers, the cytoskeleton, to the centromeres. Once the chromosomes are separated into their appropriate daughter cells, they then elongate back to the thin, long configuration found inside the nucleus. It is sometimes difficult to understand how chromosomes can change in size. The DNA itself does not change in its twisted double helical shape. It's the winding of the chromosomes around the histone proteins and how they then wind around each other that is important. Here in panel A, we have a mitotic chromosome, the most condensed form of chromosome that can exist. And in panel B, we have a chromosome in which the chromosome has been treated with an enzyme which releases some of these proteins. The DNA becomes unwound and scattered into space. So we can have chromosomes which are relaxed and those that are tightly wound. In the most relaxed condition, DNA looks like beads on a string, like a pearl necklace. Here we can see the DNA itself as a thin thread being linked together by these beads. The beads are in fact nucleosomes. At the next level of compactation, winding, the nucleosomes come together to form a spiral pattern, therefore shortening the chromosome by an order of magnitude. So these things are known as the 30 nanometer fibers, whereas this is known as the beads on a string, nucleosome. If we combine the knowledge that we've just learnt and apply that to how DNA can be accessed while still wound around its proteins, this provides us with an insight into how DNA regulation takes place. The composition of nucleosomes was deduced by experimentation. The scientists began with relaxed DNA in the form of beads on a string. They then treated that DNA with a special enzyme known as a nuclease. The nuclease is able to bind to DNA which is not associated with proteins. So in this case, the parts of the DNA in red, that is called linker DNA. So that DNA is destroyed, converted to nucleosomes, and released into the test tube. The remaining DNA is now tightly associated with the nucleosome structure. Using other techniques, the scientists were able to remove the DNA from the nucleosome and determine the size of the DNA fragment, which turned out to be 147 base pairs in length. The remaining particle, the nucleosome itself, was further disassociated using additional techniques, and its structure was determined to consist of 8 subunits. Those subunits translate to 2 H2A polypeptides, 2 H2B polypeptides, 2 H3, and 2 H4, the total number being 8. Thus elucidating the structure of the nucleosome core particle and the amount of DNA associated with it. Incidentally, we now can combine our knowledge of the structure of DNA with the structure of a nucleosome. We learnt earlier in chapter 2 that the DNA double helix is 2 nanometers in thickness. When that DNA double helix is wrapped around a nucleosome core particle, the size is 11 nanometers. We call this the 10 nanometer fiber for convenience. Using further technological refinements, we now have a better understanding of the nucleosome core. Here we can see the DNA in gray wrapped around the different subunits of the histone proteins, each indicated by a different color. The first view shows the nucleosome from the side. The second view shows the nucleosome from below. The legend of this figure makes reference to the green structure in particular. This is a tail which belongs to one of the histone polypeptides, in this case H3. The tail is significant because it is available to be modified by enzymes in the environment, therefore changing its configuration. Each and every histone in the nucleosome core has its own tail, and these tails are different from each other, being made of different amino acids. They are therefore subject to different types of modification, and in the way that they come together to form new nucleosomes. Panel B shows us the configuration adopted by histone H2A, where the different domains form these cylinders which are important in the final construction of the nucleosome. Panel C shows us how H2B histone interacts with H2A. This is important and illustrated in the next slide, which shows us how these polypeptide subunits are brought together to form new nucleosomes. The order in which the various histones come together has been deduced. Histone H4 and histone H3 first associate in the nucleoplasm to form H3-H4 dimer. Those dimers then pair up with another dimer to form a H3-H4 tetramer consisting of 4 subunits. Meanwhile, in another location, H2A and H2B form a dimer. The dimer then associates with the H3-H4 tetramer, eventually forming the octamer consisting of 8 histone subunits. The DNA molecule really is amazing. Not only does it carry information coding for amino acids, as well as switches at the beginning of genes and information about introns and exons, it also carries information directing it to associate with nucleosomes. One such finding is that the region of the DNA helix, which makes contact with the nucleosome core, is rich in certain dinucleotides, whereas other regions which do not make contact with the nucleosome core are rich in alternative nucleotides. The book talks about chromatin remodeling, and I have found these figures from a companion textbook which illustrate this to a better degree. Seen here is a single nucleosome core particle with its associated DNA plus some linker DNA to each side. What the first figure says is that a region of the DNA which may be important for binding of proteins is tightly held to a nucleosome core and thus is inaccessible to those particular proteins, known as transcription factors. If this was to remain the case, that gene could never be turned on because it could never be accessed. However, under certain conditions, which we're still deciphering, the nucleosome core can be disassociated from a loop of the DNA. And then after a certain period of time, the nucleosome could rewrap itself around the DNA fiber, restoring the condition to its original configuration. This tells us that the nucleosome core is in dynamic flux with the DNA. This is known as the dynamic wheel concept. However, in the presence of different proteins indicated in green, the association of DNA with histone can be modified such that the green protein may ask the DNA to be released from the nucleosome core and then associate directly with that. In this case, the green protein would be the transcription factor that we just spoke about. An alternative method to nucleosome unwinding is nucleosome sliding. Here the DNA around the nucleosome is simply pushed forward or backwards by a special protein known as an ATP-dependent chromatin remodeling complex. It consists of many subunits, but uses ATP energy to relocate the nucleosome along the length of the DNA. In this way, regions of the DNA can be spun to the left or the right to become linker DNA, therefore revealing them to alternative proteins. The basic 10 nanometer fiber, the beads on a string, can be wrapped around with adjacent nucleosomes to form the 30 nanometer fiber. Research into these structures suggests that the configuration adopted by the 30 nanometer fiber can range from a loose configuration as indicated on the left side of panel C to a tighter configuration as indicated on the right side of panel C. The lower orders of DNA folding are indicated in this summary slide. So panel A is the beads on a string, 10 nanometer fiber. Panel B shows us how the 30 nanometer fiber has a loose configuration and how that can be tightened up to form a more compact configuration. One can see that access to the DNA is more readily available in this configuration and this configuration than it is in the final configuration indicated here. Returning back to the tails of the histone proteins, each nucleosome will have 8 tails because of the configuration of its core. Each of these tails is subject to environmental modification. As indicated here, a single nucleosome core particle and its associated DNA suggests how the tails are aligned in that particular configuration. It is the interaction of the tails between adjacent nucleosomes which can dictate whether that structure has a loose configuration or the compact configuration. In addition to tail interaction causing local configuration changes in the arrangement of nucleosomes, there is one other component. This is histone H1, which we have not spoken much about so far. Histone H1, indicated in green here, has the ability to insert itself on the outside of the nucleosome core particle between the DNA wrapped around the nucleosome and the linker DNA. What it does is it pulls the DNA closer, the linker DNA closer to the nucleosome, therefore causing a more tight wrapping of the DNA around the nucleosome. The impact of the configuration of DNA in regions of the chromosome is illustrated by this example here. Fruit flies, which are invertebrates, those that hang around bananas, have been studied as genetic model organisms for many decades. On occasion, some weird mutations are seen in the coloration of the eyes of such wild-type flies. The normal wild-type eye is a uniform red color, whereas in these special mutants, you have a mosaic of white and red color. What is the cause of this mutation? It appears that it's related to what we have just been discussing. The name for DNA which is tightly wound is called heterochromatin. DNA which is loosely wound is called euchromatin, EU chromatin. In this experiment, the red eye is caused by the normal product of the white gene. Whereas if the white gene is inactivated by mutation, it results in white patches. Here the effects of heterochromatin can be analyzed. In the top row, the eye is wild type, fully red without any white patches. This is because the white gene is a long way away from heterochromatin, and there is a barrier which prevents the tight packaging of heterochromatin from spreading into the loose packaging where the gene resides. In some flies, there is a chromosome inversion, a translocation of types. Here, a piece of the chromosome has swapped places by flipping over. Now the white gene has been relocated close to the heterochromatin, and at the same time, the barrier has been moved to the far side. In some cells, the heterochromatin starts migrating towards the left into the white gene, inactivating the white gene, and this causes those segments of the eyes of the fly to be white. This indicates clearly that heterochromatin is dynamic in its nature and can spread to neighboring regions of the chromosome unless prevented by special barriers made of special interactions between other proteins. Further investigation has revealed that certain amino acids in key positions in the tails of the histones can be modified by enzymes in their vicinity. One of those is lysine. Lysine acetylation and methylation are important reactions which can change the shape of the tail and its interaction with the environment. Another amino acid also present in tails is serine, and serine is prone to phosphorylation to a higher degree than many other amino acids. So here is serine with its side group OH, and that can be then phosphorylated into phosphoserine by special enzymes. When one examines tails in different states of modification, a map as presented here can be generated. So we can see at the very top, histone H2A has a tail which is reasonably long, and it can be modified at these 5 positions, which are quite common, in addition to a couple of positions somewhere else. Likewise, H2B, the alternative histone, has a far greater range of amino acids in its tail which have been known to be modified. H3 has even more locations than either of H2A or H2B in the way that it can be modified in various combinations. And H4 has a similar number of modifiable locations. Taken together, you can see that there is a wealth of modifiable locations and patterns of modification that can influence the behavior of the nucleosome core. The chapter concludes by looking at some unique ways in which genes and DNA interact. The classic way that we have been taught about genetic inheritance is that the information carried by the nucleotides along the length of the DNA double helix are important in inheritance and the way that we gain information from our parents and pass on to our children. Indicated on the left side is the effects of classic mutations. If we have a gene that's working and a DNA sequence change occurs within that gene, therefore mutating the gene and turning it off, that would then be inherited by all the descendants of that cell, including future progeny, i.e. offspring. As we witnessed by watching the Nova Science video, epigenetic inheritance has been uncovered. In this form, the DNA bases are not modified. However, the structure of the molecules surrounding the DNA information, the histones and the DNA backbone, are subject to modification. Those modified changes can then be passed on to descendant cells, where they can be altered or reversed, as indicated here. As the video alluded, it may take some regions of the chromosomes several generations before the effects can be countered.